this video, I'm going to be covering all of the extra triple science content for Module 6, Inheritance, Variation and Evolution. And if you want some free flashcards to go with this extra content, click the link in the description, fill in your details and we'll send them over to you immediately. Also in the description, you'll find a link to my resources. We have everything covered for Module 6, as well as all the other modules and all of that triple science content. In the description, you'll find another link to access our exam masterclasses. Now, there is still time to join. If you want to revise with us the night before paper 1 and the night before paper 2, click that link now and secure your spot. Right. Let's get started with the video. Okay, so this is the triple only or the biology only content from Module 6. So let's get started. First of all, we have to recap the differences between sexual and asexual reproduction, because if you're taking separate science, you've got to know the advantages and disadvantages of each type of reproduction, or the pros and cons, right? So let's just recap sexual reproduction first. Sexual reproduction obviously involves the fusion of gametes. So for example, the sperm fusing with the egg cell in animals, or the male gamete, which is pollen in plants, fusing with the egg cell or the ovule in plants. So obviously it involves two parents, it involves gametes and fertilization. We know that gametes are made by meiosis, so gametes will all be genetically different to each other. So sexual reproduction leads to genetic variation in the offspring. Now, what are the advantages of this? Well, it means that there will be genetic variation in the offspring. All the offspring will be genetically different. And this is an advantage because it does increase the survival chances of that population. For example, if there's a change in the environment, maybe a new disease is introduced, for example, variation will give a survival advantage because there is genetic differences in the organisms in that population. It means some may be better adapted than others, and those that are best adapted will survive and reproduce via the process of natural selection. So it increases the survival chances of the population. It makes it less likely that they will all be wiped out or all be killed if the environment changes. For example, like I said, if there's a new disease. Sexual reproduction in plants, when the pollen grain fuses with the female egg cell or the ovule, that produces seeds, and there is an advantage for plants in using sexual reproduction and producing seeds. Seeds can be dispersed, whether that be by the wind or by animals, and that means that sexual reproduction gives them an advantage because their seeds can travel quite far, land in the soil quite a distance away. So plants that use sexual reproduction are able to colonize or grow in new areas. Now, let's think about some disadvantages. Obviously, with sexual reproduction, two parents are needed. So, for example, animals must find a mate to reproduce with. Plants rely on pollination from insects or the wind. So this does increase the time that sexual reproduction takes, and it increases the amount of energy required as well. So as a result, sexual reproduction is more time consuming and more energy demanding than asexual reproduction. So to summarize, big advantage: introduces genetic variation, increases survival chances, but disadvantages: it's more time consuming and it does require more energy. Let's compare sexual reproduction to asexual reproduction then. And again, we'll just quickly recap what asexual reproduction is. With asexual reproduction, there's no gametes involved, so there's no fertilization and no meiosis, because meiosis only makes gametes. There's no gametes involved, no fusion of gametes. Only one parent is required, and this means the offspring are getting all of their DNA from just one parent, which means the offspring will be genetically identical to that parent and also to each other if they're coming from the same parent. So the offspring will be genetically identical. We can use the term they'll be clones. That means genetically identical. So advantages of this: well, this only requires one parent, as we said, so it's a faster process or less time consuming than sexual reproduction, and it requires less energy than sexual reproduction. So in plants, they don't have to wait to be pollinated, they don't have to wait to flower, they don't have to wait for seeds to be formed. In animals, they do not need to find a mate to reproduce sexually. So it's faster, it's less demanding of energy. You get many genetically identical offspring. Now, you could give that as an advantage because these offspring can be produced quickly if conditions are favourable. And that means that all the offspring, because they'll be genetically identical, if the parent had an advantageous trait, the offspring will inherit the same advantageous traits as the parent because they'll be genetically identical. So if the parent plant had large, tasty fruits, I don't know, a strawberry plant with big, red, juicy, tasty strawberries, then the offspring will inherit that same advantageous trait because it has the same DNA or the same genetic material. Disadvantages, though, obviously with this type of reproduction, there is no genetic variation. The offspring are genetically identical. So all the offspring are potentially susceptible to the same disease. So if the environment changes, all the offspring could be killed. If there's a new disease introduced, all the offspring could be killed. So there's less chance of survival because they don't have that genetic variation. So they don't have the opportunity to undergo natural selection where the most suited to the environment will survive and reproduce. They're more likely to all be killed by a severe environmental change. Okay, now we've done the pros and cons of sexual and asexual reproduction. The next thing for triple scientists to learn in Module 6 is a little bit more about the structure of DNA. Now, again, we're going to start with some revision because there is some content here that everybody has to learn. So we know that DNA stands for deoxyribonucleic acid. We should all know it's a polymer. That means it's made from many repeating monomers. It's a long chain of monomers. The monomer in DNA is called a nucleotide. We should also already know that DNA is double-stranded, so there are 2 strands, and we should all already know that it has a double helix shape. So that's kind of the information that everyone needs to learn, right? It's a polymer, it's made of many repeated monomers, it's double-stranded, and it has a double helix shape. We also know that in a eukaryotic cell, it's found inside the nucleus, and we know that it carries the genetic instructions for making proteins. But if you're triple science, we need to know a little bit more. So let's have a think about the monomer. I'll show you a picture of it now. So this is the monomer, or the single unit from which DNA is made. But obviously DNA is a polymer, and it's double-stranded. So I need you to imagine 2 strands of many of these nucleotides, but you could be asked to name the component parts of a nucleotide or label a nucleotide. So this basic unit of DNA consists of a phosphate group, which is shown here. We represent that as a circle. So that's a phosphate group or a phosphate ion. It also has a sugar. It's actually called deoxyribose sugar. It has 5 carbons, that's why we represent it with a pentagon shape, and it has a base. Now the bases, we can use the letters to represent the bases. So there are 4 different bases, which gives us 4 types of nucleotides. The 4 bases are A, T, C, and G. I'll just tell you quickly what that stands for, but remember you can just use the letters. So the A is for adenine, the T is for thymine, the C is for cytosine, and the G is for guanine. So 4 different types of bases, but all nucleotides will have a base, one of those 4, plus a sugar, plus a phosphate group. And then you need to imagine that DNA is a polymer, right? So I'll just draw a few so you get the idea. So it's made of many of these nucleotides joined together, and it's double-stranded. So there are actually 2 strands in the DNA. The bases are in the middle. Oh, I'm going to run out of space. The bases are in the middle like this. There you go. Look at me doing it upside down. So the bases are here in the middle, and then the sugars and the phosphates are on the outside. So we say that there are 2 sugar phosphate backbones. That's what forms those 2 strands. Now, if we delve a little bit deeper into the structure of DNA, so you can see here, this is kind of what I was trying to draw before. We've got the 2 strands. They'll be made up of the sugars and the phosphates. We can see it's twisted into a double helix shape, and you can see the bases are in the middle. So let's just go through that again. So we know it's a polymer. It's made of many monomers, and we know the monomer is called the nucleotide. So it's a polynucleotide. It's made of many nucleotides. It has 2 strands. The 2 strands form our double helix structure. There are sugar and phosphate groups in the backbone. So the 2 blue strands here are representing the sugars and the phosphates. And then the bases stick out and are in the middle of the 2 strands, in the middle of the double helix. Now, there is a base pair rule that we should know. So the bases pair up in the middle, as you can see here, but an A base, which stands for adenine, always pairs with a T base, which stands for thymine. So adenine and thymine are always together, and a G base, which you can see here, always pairs with a C base. So adenine and cytosine are always together. This is known as the complementary base pair rule. Basically, A and T are complementary in shape, so they bind together, and G and C are complementary in shape, so they bind together. Now, there's ways to remember this, like at the golf course, at the group chat. Some students remember that the straight letters pair together and the curly letters or the curvy letters pair together, but you do need to know that complementary base pair rule. Now, some more definitions that are really important in Module 6. We should be able to define a gene. Now, a gene is just a small section of DNA that will code for a protein. But if you're triple science, you need to know a little bit more than that. The reason that a gene or a small section of DNA codes for a protein is because the order of the bases in the DNA will determine the order of amino acids. So you can look at kind of this strand here, right? Let's look at this strand. So you've got a thymine, you've got a guanine, you've got an adenine, you've got a cytosine. The order of those bases determines the order of the amino acids that make up the protein. And that's why we say a gene is a section of DNA that codes for a protein. And the way it works is 3 bases in DNA provides the code for 1 amino acid. So those 3 bases there that I've just circled, they will provide the code for 1 amino acid. And then the next 3 bases provides a code for another amino acid. And then the next 3 bases provides the code for another amino acid, and so on. So 3 bases codes for 1 amino acid. Now we call these 3 bases a triplet. So you can say a triplet is 3 bases in DNA that provides the code for 1 amino acid. So it's a triplet code. It works in sets of 3. So for example, if you had 12 bases in your DNA, you might be asked how many amino acids does that code for? Obviously, it would code for 4 amino acid because 12 divided by 3 equals 4. There are 4 sets of 3. There are 4 triplets. So it would code for 4 amino acid. Overall, it's really important that we remember this sentence. I said this to my students very recently. The order of bases in DNA controls the order of amino acids to form the protein. That is how DNA is acting as your genetic material. It codes for proteins because the order of bases determines the order of amino acids in a protein. Okay? Right, bearing that in mind then, let's think about protein synthesis. Now, protein synthesis just means literally making proteins. It's not as complicated as many teachers make out. We're going to make it nice and simple. So protein synthesis is the process by which cells make proteins. And obviously we already know from paper 1 that proteins are made of amino acids. They're also polymers, right? They're made of many amino acids joined together. But to make these proteins, we're going to follow the instructions that are carried by DNA because DNA has bases. The order of bases is what's going to determine the order of amino acids in the protein. Now, DNA is obviously found in the nucleus, We need the DNA to act as the code because the order of bases in DNA, I've said this so many times now, it determines the order of amino acids in a protein. And we also know that it's 3 bases or a triplet that will code for 1 amino acid. Now, DNA is in the nucleus, but we need the code to get to the ribosomes. You can see a ribosome here in purple. Ribosomes are found in the cytoplasm, right? The DNA is in the nucleus. But proteins are made at the ribosome. So the amino acids are going to be put together to make the proteins or synthesize the proteins at the ribosomes. So we need to figure out, well, how is the genetic code that is stored in the DNA, which is in the nucleus, how are we going to get that information to the ribosomes so the ribosomes can put the order of amino acids together and build the protein correctly? So let's go through the process of protein synthesis, and this is much, much simpler at GCSE than it is at A level. So let's try and keep it simple. So DNA remains in the nucleus. It can't leave the nucleus. It's too big. There's too much of it. It stays in the nucleus, but that provides the genetic code. So what we have to do is make a template. We have to make a copy of the gene. By making a template, the template can move out of the nucleus and it can go to the ribosomes, which are in the cytoplasm. Now, this template molecule, your teachers might have told you the actual name of it. The template molecule is called messenger RNA. But you don't have to call it messenger RNA at GCSE. You can just call it a template molecule. But hopefully that makes sense so far. The DNA is in the nucleus. We need the code to get to the ribosomes, which are in the cytoplasm. So we make a copy of the DNA. We make a template molecule called messenger RNA, which can leave the nucleus and carry the genetic code to the ribosome. Now, once we're at the ribosome, we need the amino acids to be brought to the ribosome, right? So this involves carrier molecules. Carrier molecules bring specific amino acids to the ribosome. Each carrier molecule doesn't just carry any old amino acid, it carries a specific amino acid, and they are kind of matched to the template molecule to ensure the amino acids are joined in the correct order. Okay, let's have a look at this on the diagram. So this is our template molecule, which if you want to, you can call mRNA or messenger RNA. This is made in the nucleus. It's just a copy of the DNA code, right? Because the DNA can't leave the nucleus, but the template molecule can. So the template molecule is a copy of the DNA code. As you can see on the diagram here, it's going to go to the ribosomes where the proteins are synthesized, and the carrier molecules are going to bring the amino acids, the specific amino acids to the ribosome. And the amino acids, as you can see, are going to be joined together to make the protein. Now, the carrier molecule is known as tRNA or transfer RNA. But again, you can just call it a carrier molecule. You don't have to learn tRNA because it says carrier molecule on your spec, right? But we do need to know that the template molecule carries the genetic code to the ribosome, and the carrier molecules bring the specific amino acids to the ribosome, and the amino acids get joined in the correct order. So the ribosome joins the amino acids together in the correct order, and that's what forms the protein chain, the chain of amino acids. After the chain of amino acids has been formed, it will fold to create a unique 3D shape. Each protein has a unique shape, right? A unique 3D shape. And that shape is going to help the protein to perform its specific function. Now, you've already learned about lots of proteins on your specification, whether you've realized it or not, right? Enzymes are proteins, hormones are proteins. We have structural proteins such as collagen or keratin or elastin. We even have transport proteins like hemoglobin, right? That transports oxygen in our red blood cells. But all of these proteins rely on having a specific shape. Let's take the enzyme example as the most obvious one. Enzymes have to have a specific or unique 3D shape because the shape of their active site has to be complementary to their substrate, right? So the substrate can bind to the active site, and so we can form enzyme substrate complexes. So the shape of the protein is really important for the protein to be able to carry out its function well. And obviously all of these proteins are made at the ribosomes. We will be making sure the order of amino acids is correct. The order of amino acids will be based on the sequence of bases in DNA. So the sequence of bases in DNA determines the order of amino acids in the protein. And the order of amino acids in the protein will ultimately determine how it folds, which will determine its final complex 3D shape. So let's link this all together then. So how does DNA structure, or the order of bases in DNA, affect the protein? I'm going to say it one more time so we definitely remember this sentence. The reason DNA is linked to protein is because the order of bases in DNA determines the order of amino acids in a protein. And we know that each triplet, or each set of 3 bases, codes for 1 amino acid. What this means is if there's a change in the DNA base sequence, which we call a mutation is a change in the order of bases. If we change the order of bases in DNA, and that could happen randomly, right? Mutations are random. We change the order of bases in DNA, that would change the order of the amino acids in the protein. The amino acids would be put together in a different order. That could affect the overall shape of the protein, which could affect the overall function. In fact, it could mean that the protein cannot function. It's a non-functional protein. Now, not all mutations have this effect, just to be clear. Many mutations will have no effect on the overall shape of the protein and no effect on protein function. We call them silent mutations if you're interested. But some mutations could lead to a change in the order of amino acids, a change in the folding of the protein, a change in the shape of the protein, potentially meaning that the protein is non-functional. Okay? So just bear that in mind. It doesn't always lead to a non-functional protein. It might have no effect on the protein. Some mutations as well can be positive because you could get a mutation, which is a change in the order of bases in DNA. That could lead to a new allele, so a new version of a gene, which actually gives an advantage. It might actually affect the phenotype in a positive way. It might give the organism an advantage to have that mutation and that new allele. And that could actually increase chances of survival and lead to natural selection. Okay, so there's actually kind of 3 outcomes of a mutation. Could be positive, give them a survival advantage, could have no effect whatsoever on the protein, no effect on their phenotype, or it could have a negative effect where it changes the order of amino acids, changes the shape of the protein, protein is non-functional, and that could have a negative effect on the organism's phenotype. Okay? Right. What else do we have to do if we're triple science? We have to do a little bit more about inheritance. We have to look at someone called Gregor Mendel. Now, Gregor Mendel was not actually a scientist. Gregor Mendel was a monk. And in the mid-1800s, he did loads and loads of experiments on pea plants, lots of breeding experiments on pea plants. And we call him the father of modern genetics because even though we didn't know about DNA or genes or alleles back then, with his experiments, he discovered that characteristics are inherited in clear and predictable patterns. So he noticed that characteristics were being passed on from the parent pea plants to the offspring pea plants, and he started to notice patterns in inheritance. He proposed that units, that's what he called them, units were passed on from parents to offspring. We obviously now call these units genes, but remember back in the mid-1800s, we didn't know about DNA or genes yet, so he was calling them units. Units are being passed on, characteristics are being passed on. Now we know it's genes that are being passed on. Now, his work was not recognised until after his death, but we do call him the father of modern genetics. It was later on, so in the late 19th century or the late 1800s, that chromosomes were observed during cell division as microscope technology improved. And it wasn't until the mid 20th century or the mid 1900s that we determined the structure of DNA. It was Watson and Crick and Rosalind Franklin that actually developed our model of the structure of DNA. That's when we also worked out kind of how genes worked, the mechanism of inheritance. But Mendel, back in the mid 1800s, before we'd discovered DNA structure, before we'd identified chromosomes and genes, he actually did manage to figure out that some units, as he called them, were dominant and some units were recessive. We now call them alleles, right? Different versions of genes are dominant and recessive. But he noticed that long before the discovery of DNA. So let's have a look at an example. And kind of everybody needs to know this, whether you're combined or separate science. So he was crossing plants. Now imagine you're crossing like a pure breeding red plant. So a homozygous red plant, crossing that with a purebred white plant. So a homozygous white plant. He noticed that all of the offspring were red. Now, he didn't refer to it as a dominant allele, but he noticed that the red trait was dominant, right? Even though the parents were red and white, all the offspring ended up red from that first cross because red must be dominant, a dominant unit. Then when he crossed those plants, I'll do a little Punnett square at the side so you can see the possible outcomes. He crossed those plants and he noticed that some of them were red, and some of them were white. So he then noticed that if you cross two parents that are heterozygous or have a dominant and a recessive allele, then you'll start to see offspring with the recessive trait in that next generation. He called this the F1 generation, like the first generation, and this was the F2 generation, the second generation. And he started to see this 3-to-1 ratio of phenotypes in the F2 generation. So even though he didn't use the term gene or allele, he did manage to kind of conclude that units are being passed on from parents to offspring. Some units are dominant, will always be expressed. Some units are recessive, and obviously we now know if it's caused by a recessive allele, the characteristic will only be expressed if the offspring inherits 2 copies of the recessive allele. Now, the next thing you need to know about Mendel is why did people not really accept Mendel's theories of inheritance? Why was his work not recognised until later after he died? So answers you can give to a question like this in an exam, you can say he was only a monk, or he was not a scientist. So other scientists didn't really pay much attention to his work, didn't really peer review it, didn't really do their own experiments to back it up and support it, right? So he was only a monk. Don't just say he was a monk. You have to convey the idea that he was only a monk, he was not a scientist. He carried out most of his research on pea plants. So I suppose scientists weren't sure whether you could generalize his findings to other organisms and other species because he mainly just worked on pea plants. His work was published in an obscure journal. So it was published, but it was more in like a gardening journal as opposed to a scientific journal. So again, not many scientists read his work, didn't carry out experiments to provide additional evidence, didn't back it up, didn't peer review his work. And another really good one to give, he couldn't really explain the science behind why characteristics were inherited. You can say we had not yet discovered DNA, we had not yet discovered genes, or we had not yet discovered the mechanism of inheritance. Like exactly how these units are being inherited and how it all works, right? We didn't know the structure of DNA. We didn't know about chromosomes, genes, alleles, all that stuff. So we didn't know about the mechanism of inheritance, which made it very, very difficult for him to explain the science behind what he had found, okay? So try to make sure you remember at least 2 of these. Maybe this one and this one are the easiest to remember. It's up to you. Try and remember 2 of them. Don't mix these answers up with your Darwin answers. So there's another similar question, isn't there, about why people didn't believe Darwin's theory of evolution by natural selection. And the main answer we give for that one is it went against people's religious beliefs and he didn't have enough evidence. These are different answers. Similar, but different. So for Darwin's theory of evolution, we say people didn't believe him because he didn't have enough evidence. It went against their religious beliefs. Whereas for Mendel, we say he was only a monk, not a scientist. His work was published in an obscure journal, not a scientific journal. He only carried out his research on pea plants. Okay. This last one here is the only one, the only answer that you could give for both Mendel and Darwin. That is true of both scientists. You can say that for both. Neither of them understood the mechanism of inheritance yet, so it was hard for them to explain their findings. Okay. The next thing we're going to do is plant cloning. Now, plant cloning is a process of creating genetically identical copies, obviously producing clones of a plant. So they'll be genetically identical, they'll have the same DNA as the parent plant. So it's a type of asexual reproduction. It's a way of cloning a plant. It ensures that the offspring inherit the same desirable traits as the parent plant. So it could be that the parent plant grows really quickly, grows to a large size, so there's a large yield. It could be the plant has bright coloured flowers or nicely scented flowers or produces large tasty fruit. Obviously, it depends on the plant. It might be the plant is more pest resistant or disease resistant, but you might want to clone a plant with desirable characteristics because then you're guaranteed the offspring will have the same desirable characteristics. Now, there are two methods that we need to study for plant cloning, two ways we can do it. The first one is called tissue culture. Now, tissue culture, as the name suggests, a tissue is like a group of cells, right, that carry out a particular job. But tissue culture basically involves using a small group of cells from part of the plant to grow a whole new plant. So here we've got the plant, the parent plant that we wish to clone. They've removed a leaf from the parent plant and they've scraped off a few cells, and you're using just a small group of cells or a few cells to grow a whole new plant that will obviously be genetically identical to the parent plant because there's just one parent, it's asexual reproduction, it's got all of its DNA from one parent. So you scrape off the few cells. The cells are grown in sterile conditions in a nutrient-rich jelly called agar. Agar jelly is basically a growth medium. It provides the cells with all the nutrients they need, and you can add hormones to the agar jelly, such as auxin, which is a plant hormone you should know, to stimulate growth. Okay, and obviously keeping it in sterile conditions, so there's no bacteria, no fungi, not going to cause any infections to the plants as they grow. But yeah, you provide it with the nutrients it needs to grow, such as, I don't know, amino acids, mineral ions. You provide it with auxins to stimulate growth, and those plants will start to grow into teeny tiny little plants that we call plantlets. Once they're big enough and strong enough, they're transferred into soil, which obviously you need to water. The soil is moist. They'll continue to grow, and then you'll get a whole new plant from just a small group of cells from the parent plant. And the new plant will be genetically identical to the parent plant from which the cells were taken. Now, if we look at the second method is called taking cuttings. This is a more simple and traditional method that you could even have a go at doing yourself, or you may have done in school. This is simply where a section of a plant is cut from the parent plant. So you can see that here. We are cutting off a small portion of stem with a few leaves, making sure that obviously we leave the parent plant with some leaves as well, so the parent plant doesn't die, isn't too damaged. So leave some leaves on the parent plant so it can continue to photosynthesize, but take a portion of shoot or maybe a side shoot from the parent plant. We do cut it at an angle because that increases the surface area of the cut stem, so it should be able to absorb more water. We also dip the cut end of the stem in rooting powder, which again contains hormones such as auxin to stimulate root growth. It's then potted into soil, which again you would need to water to moisten the soil so it has enough water for photosynthesis. Now you might get a question where you see a transparent or a clear kind of polythene bag placed over the growing shoot. Now the reason for that, it keeps it a little bit warmer, so it helps to increase the temperature. It also keeps pests off of the growing plant, but also it traps water vapor in here, right? As water evaporates from the leaves of the growing plant, that water vapor will be trapped in the bag. It will make it nice and moist or nice and humid in here, which will help to reduce water loss by transpiration. So there's a few reasons you could give for the benefit of that. Yes, it keeps away pests. Yes, it makes it a little bit warmer because it acts like a mini greenhouse, increases the temperature, but it also makes the environment in here really humid or really moist, which will reduce the rate of transpiration or reduce water loss by evaporation. Obviously, again, this is an example of plant cloning. It's an example of asexual reproduction because you're just using one parent plant. So the offspring plants will again be genetically identical to the parent plant from which the original shoot was taken. Now let's go through the pros and cons of each method. So in terms of tissue culture, which was, remember, where we just scraped off a few cells and used the cells to grow whole plants, offspring plants are genetically identical to the parent plant. So if the parent plant had desirable traits, you're guaranteed the offspring plant will have the same desirable traits. That's kind of a pro for both methods though, whether it's tissue culture or taking cuttings. You can give that as an advantage. But with tissue culture, you can produce many offspring plants from one parent plant. You can also say it's much faster because you only need a few cells. So you can have one parent plant with desirable traits that you wish to clone, and you can produce many, many offspring plants from that parent plant really quickly. And another advantage of tissue culture, there will be less damage to the parent plant because you only need a few cells. Whereas with taking cuttings, you obviously need to cut off a whole shoot, side shoot. So there's less damage to the parent plant with tissue culture. If you think about taking cuttings though, what are the advantages of that? Again, same advantages with tissue culture in terms of the plants will be genetically identical to the parent plant. They will have the same desirable traits. It's cheaper to do, it's easier to do. And compared to growing plants from seeds, which have been formed with sexual reproduction, it's quicker than growing plants from seeds because you're starting with a shoot already with a few leaves. So it's quicker than growing plants from seeds. It's cheap, it's easy to do, but you can't make as many offspring from one parent plant as you can with tissue culture, and there will be more damage to the parent plant than there was with tissue culture. If we look at these together and kind of summarize in general the pros and cons of plant cloning, I mean, both of these methods can help to preserve endangered plant species. So if a plant is at risk of becoming extinct, a plant species is at risk of becoming extinct, you can use these methods to preserve that endangered plant species. With both of these methods, you're going to make sure that your offspring plants have the same desirable characteristics of the parent plant, whatever that may be. And it can be done year round, so you're not reliant on the seasons. You don't have to wait until spring when the temperature's warmer or there's a greater light intensity. You could do this year round if you provide the optimum conditions for the growth of your plants. In terms of risks, these apply to both methods. All the clones will be genetically identical to the parent plant, no genetic variation, which does make them more vulnerable to disease. Because if the parent plant was more vulnerable to disease, all the clones will also be more vulnerable to disease, so they could potentially all be killed by a particular disease. It reduces genetic diversity. We know that genetic diversity is important for survival because if you've got genetic variation, it means some plants would be more likely to survive, reproduce, pass on their genes. If we've got reduced genetic diversity, you don't have as much scope for natural selection. So they could potentially all be killed if the environment changes, or all be killed by a new disease. So reducing genetic diversity or just creating genetically identical plants is not good for survival of the population in general. Then we have to look at animal cloning. And again, there are two techniques we've got to study. So we're going to go through both. Animal cloning is the process of producing genetically identical copies, but this time of animals, not plants. Again, it uses asexual reproduction techniques. We're ensuring that the offspring will have the same DNA. Now, this first one, just to clarify, this first one is called embryo transplants. And actually with this one, the offspring will not have the same DNA as either parent. Just to clarify, that's a little bit misleading, right? The offspring with this technique will not be genetically identical to the parents. They will be genetically identical to each other, but not to either parent. I'll explain more about that in a second. So this first one is called embryo transplants, or sometimes we call it embryo splitting. In fact, I've seen it called embryo cloning as well. It's all the same thing. So what we're doing here is we're taking an egg cell from a female animal. We've got a female cow here. We're taking sperm cells from the male animal of the same species. So we've got sperm cells from a bull here. We are allowing fertilization to take place in a test tube or in a petri dish. So this is in vitro fertilization, not inside the body, but in the lab. Obviously, when fertilization occurs, an early embryo will start to develop. So we'll have a fertilized egg cell, or a zygote, as we call it. The zygote will divide by mitosis, just basic cell division for growth, and it will start to form an embryo, a cluster of cells. But when the embryo is still in the early stages of development, the embryonic cells are separated or split apart, which is why we call it embryo splitting. And we're splitting the embryo cells apart at an early stage of development when they haven't yet specialized. They're still stem cells, right? They're still embryonic stem cells. So we separate the cells from that embryo, and those cells will continue to divide by mitosis, normal cell division for growth. So the embryo cells will continue to divide by mitosis, and now you can see we've basically created 4 separate embryos from one original embryo. So we created one original embryo, we separated the cells at an early stage of development. Now we've got 4 embryos from the same embryo. Those embryos are then implanted into the uterus of host mothers or surrogate mothers. The embryos will continue to grow into a fetus, and then we'll get genetically identical animals or clones being born. Now note, with this method of embryo transplants or embryo splitting, the offspring will be genetically identical to each other, but not to either parent. And this is because initially this embryo was made from 2 parents, right? This embryo was made by sexual reproduction initially, because we had DNA in the sperm cell, we had DNA in the egg cell. We mixed that DNA together at fertilization. So this embryo here is not genetically identical to the male, it's not genetically identical to the female, it contains a mixture of DNA from 2 parents, right? But then we use asexual reproduction because we split the embryo into separate cells and create 4 embryos in this case from one embryo. So these embryos will be identical to each other because they all came from this early embryo, but the offspring are not going to be identical to either parent because they actually have a mixture of DNA from 2 parents. But the aim of this, obviously, if we've chosen a male with desirable traits, we've chosen a female with desirable traits. Hopefully this embryo has the same desirable traits. So we're able to get 4 offspring here from these parents instead of just the 1 offspring. So we're increasing the number of offspring with the desirable traits. We're able to get more offspring quickly with the desirable traits, and the offspring will all be clones of each other. Now, with the second type of animal cloning, we are actually going to clone an adult animal. So we are going to clone a parent here. This is called adult cell cloning, and you may have heard of Dolly the sheep as a very famous example of a sheep that was cloned from an adult sheep. Let's go through how this works. So, the nucleus is removed from an unfertilized egg cell. So here on the diagram, we've got a female sheep. We take an egg cell from that sheep and we remove the nucleus. So this is an egg cell with no nucleus. We've removed the nucleus from the egg cell. Then we take a body cell from the animal that we wish to clone. So in this example, we are cloning female A, sheep A. Because from this animal, we've taken a body cell and we've extracted the DNA, or you can say extract the nucleus, because the nucleus contains the DNA, right? Okay, so we've got a body cell from the animal we wish to clone, and we extract the DNA, or we extract the nucleus. We've then got an egg cell from any other female animal of the same species. We've removed the nucleus from that egg cell because we don't want to clone this sheep. We don't want their DNA. But we take the DNA that we got from sheep A, or the nucleus that we got from sheep A, and we fuse it with the empty egg cell. So we're inserting all of female A's DNA into an empty egg cell. So this egg cell will have all of the DNA from one parent. It will have all of female A's DNA. We give that fertilized egg cell an electric shock, which stimulates division. Obviously, this would now be mitosis for growth. It will start to form an embryo, which we can see here, a cluster of cells, and then the embryo is inserted into the uterus of a female host mother or surrogate mother, where it continues to develop. And the offspring, in this example on my slide, will be a clone of sheep A, because all of the DNA came from sheep A. Okay, let's just go through it again to try and simplify it. So you need an egg cell, and you remove the nucleus from the egg cell. You discard it. You take a body cell from the animal you wish to clone, and you extract the nucleus or the DNA, and you put the DNA of the animal you wish to clone into the empty egg cell. Give it an electric shock. It will divide to form an embryo. Insert that embryo into a host mother's uterus, and the offspring will be a clone of the animal that you got the body cell from, not the egg cell, the body cell, the DNA. So note the type of cloning here, adult cell cloning, will produce an animal that is genetically identical to one parent, the parent from which the body cell was taken and the nucleus of the body cell was obtained. This nucleus contained all the DNA of this one parent. So the offspring here is a clone of an adult animal. We call it adult cell cloning because we took a cell from an adult animal, we used the DNA, we cloned that animal. Okay, so this is different to the embryo transplants where we just create an embryo and split it and the offspring are identical to each other. Here we're actually cloning an adult animal, just like we did with Dolly the sheep. Let's look at some pros and cons then of animal cloning. So benefits. Obviously, the point is to produce animals with desirable traits. So if you've got parents with desirable traits or an animal you want to clone with desirable traits, you can clone it, you know the offspring will have the same desirable traits because they're genetically identical. So you might think of traits like, well, a female cow with a high milk yield, animals that are disease resistant. I've seen questions about cows where the female cow you want to clone produces like low fat milk or creamy milk. I don't know. Do be careful though with the with this idea, because we talk about female cows producing milk, right? Some students fall into the trap of saying, choose the bull that produces the creamiest milk or the highest yield of milk. Remember, male cows or bulls do not produce milk. It's only female cows that produce milk. So just a reminder of that, to be careful. Another benefit, it can help to save endangered species to prevent their extinction, because we can produce clones relatively quickly to up their population size. We can clone animals that produce useful proteins or useful human medicines and get them to excrete them into their milk so we can collect them very easily as well. And we can use cloning techniques for scientific research. In terms of risks though, again, we've got the same kind of risk as we did with the plant cloning. Whenever you are cloning an organism, it reduces genetic variation. All the clones will be genetically identical. You are decreasing chances of survival because they could all be susceptible to the same disease and could potentially all be killed by the same disease. There's ethical concerns with this because cloning embryos, the embryo might fail to develop properly, and you can argue, embryos have a right to life and the embryo may not survive and may be damaged, etc. Low success rate and high costs. And what we found with Dolly the sheep, she didn't live as long as expected. So clones may have shorter lifespans than what you would expect, or the cloned animals may experience greater health problems. Okay. Last little bit of module 6 for triple science then. We're going to go back to evolution, quickly recap Charles Darwin, and then add on the extra bits that you need. So remember Charles Darwin. Charles Darwin went to the Galapagos Islands, did many observations, made many drawings. He's proposing the theory of evolution, right, by natural selection. In summary, he's saying that individuals in a species vary. There's many differences. The ones that are better suited to the environment are more likely to survive and reproduce, passing on their genes. That's basically natural selection in a nutshell. They pass on their genes or their favourable traits to the next generation. And this is repeated over many generations until you see the beneficial adaptation become more common in the population. So we're saying gradually over time, species change, species evolve. And it's through this process of natural selection because the best adapted survive, reproduce, pass on their genes, and then these advantageous genes become more common in the population. Now, we need to know about another theory. Now, this theory was proposed by Jean-Baptiste Lamarck. Don't have to know his first name, but you should know his surname, Lamarck. He proposed a different theory of evolution before Darwin's theory of natural selection. And it was based on two main ideas. So the first one, use and disuse, if you like. So he basically believed that if an organism used a characteristic a lot during its lifetime, it would become more developed. So for example, giraffes and their long necks. Because they are stretching and stretching and reaching to get food, they are using their neck more during their lifetime. So gradually it gets longer. Or think about flamingos and their long legs, right? Flamingos don't want to get their feathers wet because they won't be able to escape predators as easily, and they want to be able to wade in the water and get food without getting their feathers wet. So they stretch and they stretch and they stretch their legs. They're using their legs more during their lifetime. Gradually their legs become longer. And then he suggests that these acquired characteristics that the organism has developed during their lifetime can be passed on to their offspring. So going back to the giraffe example, if they stretch and stretch their neck, their neck gets gradually longer during their own lifetime, they then pass on that characteristic to their offspring. This theory was known as the theory of acquired characteristics, because basically he's saying that an organism can acquire or develop a new characteristic during their own lifetime and then pass it on to their offspring. Now, we know now that acquired characteristics, things that you develop during your lifetime, are not passed on genetically. Changes during your lifetime do not affect your DNA, right? Whereas Darwin's theory is now supported by modern genetics and DNA evidence, right? Lamarck's theory is not supported by modern genetics and DNA evidence. But it's still a theory that we learn about as an alternative, and this idea that you can overuse a part of your body, I suppose, during your lifetime, develop that body part, change that characteristic during your lifetime, and pass it on to offspring. Not supported by evidence, like Darwin's theory of natural selection is, okay? Now we need to talk about Alfred Russel Wallace. He was a biologist and an explorer. He kind of independently came up with the same idea as Charles Darwin, which we now have lots of evidence to support. So he too developed this idea of evolution by natural selection. And it was even around the same time as Charles Darwin. He basically came up with the same proposal, the same theory, the same way it happens, survival of the fittest or natural selection. In 1858, Wallace and Charles Darwin actually jointly published their ideas on natural selection or survival of the fittest. And it was this that encouraged Darwin to publish his famous book on the origin of species. And that's kind of why we all know Charles Darwin's name. We all know he's kind of responsible for this theory and natural selection. But actually, Alfred Russel Wallace had the same ideas and did write to Charles Darwin. They worked alongside together. He came up with the exact same idea. So what else do we attribute to Alfred Russel Wallace? He did some other things that are super important. So Wallace, remember his name, Alfred Russel Wallace, he also studied warning coloration in animals. So this could come up on a triple science paper. Warning coloration refers to bright and distinctive colours that some animals have, like the poison dart frog or the coral snake or a hoverfly versus a wasp, right? Warning colours, brightly coloured, brightly patterned. And he's saying they seem to act like a visual signal to predators that indicates this animal is toxic, produces a poison, maybe it tastes really bad, maybe this animal is dangerous. So he called them warning colours, right? Bright colours in nature act as warning colours. So for example, the bright stripes, you've got yellow and black stripes on a wasp, indicate the wasp will sting, it is dangerous. The vivid skin of a poisoned dart frog, brightly coloured skin, produces a poison, it's poisonous, it's toxic. It warns predators to stay away. So bright colours in certain species are signals basically to predators that that animal is poisonous or dangerous. And these colours evolved through natural selection because obviously it's an advantage, right? If the bright colour is signalling to predators, I'm dangerous, don't eat me, don't try and kill me or attack me, that animal is more likely to survive and reproduce and pass on the genes for that characteristic. So over time, populations will gradually evolve and change so that all of that species has that bright warning colour because it increases their survival chances. Now, one other example to look at here, because they've used this on the exam before, harmless species, right, that aren't toxic, aren't poisonous, aren't dangerous in any way, shape or form, may evolve to mimic these warning colours. This is called mimicry. Because by doing that, they gain protection by resembling dangerous species. So they're not dangerous, they don't produce a toxin, they're not harmful. But if they mimic or look like a species that is, that will also prevent the predators from trying to eat them, kill them, etc. So the example they've used on the exam before is hoverflies, because hoverflies mimic wasps. They have the same yellow and black stripy patterning. Other animals or predators may mistake them for a wasp. Basically, wasps sting, right? Wasps are dangerous. Hoverflies don't. But they think that the hoverfly is a wasp. They think the hoverfly would sting them. And this helps the hoverfly avoid being eaten by predators. Look how similar they look. This is called mimicry. So the hoverfly is safe, it doesn't sting, but the wasp does sting. The hoverfly has evolved by the process of natural selection to mimic and resemble the wasp. And the way that will have worked is there'll be genetic variation in the DNA of the hoverflies, originally due to mutations. Some of them might start to have some yellow patches. They survive, they reproduce, they pass on their genes, their offspring have these yellow patches. The ones that have the stripy yellow, they're more likely to survive, reproduce, pass on their genes. Gradually, over many, many generations and many years, the hoverfly population, they all now resemble wasps because it gave them a survival advantage, helped them survive, reproduce, pass on their genes for this advantageous characteristic. Okay. Russell Wallace then, very important guy. Not only did he come up with the same theory of evolution via natural selection, not only did he look at warning coloration and mimicry, but he also did lots of pioneering work on speciation, which is another triple science topic. So speciation is the process where new species evolve from pre-existing ones. So you've got an original species, it evolves into a new species, or it evolves into two new species, or three new species. Let's go through how it's done. Now, to understand speciation, you kind of need to understand these three facts. So a species, the definition of a species, is a group of organisms that can interbreed or reproduce with each other to produce fertile offspring. That is your biological definition of a species. I suggest you memorize that. Don't just say, oh, a species is a group of organisms that look the same or have the same characteristics. No, they can interbreed or reproduce with each other to create fertile offspring, as in offspring that will also be able to reproduce. Another thing we should know before we look at this more deeply is if populations of the same species become separated, they may evolve differently. And what can happen is if they change so much that they can no longer breed together to produce fertile offspring, they have become two different species. Okay, so if we separate a species, they are isolated from each other, they can evolve in different ways and become so different genetically that they form two different species because they can no longer interbreed to produce fertile offspring. So let's go through the steps of speciation. And we only look at one type of speciation at GCSE. And the type that we look at is where speciation occurs because of geographic isolation. So we are physically splitting or physically isolating a population. So that's step number one, isolation. The population that were all the same species, right? They were all breeding, interbreeding, creating fertile offspring. They have to become geographically isolated. They have to be physically separated or isolated. This could be by a mountain range that keeps them separate, maybe a river, maybe climate change leads to a flood on an island, which separates the island into two parts. Could be a volcano. Yeah, could be a motorway. I don't know. It depends on the size of the species, right? But there's got to be something that's acting as a physical barrier, causing geographical isolation. So we've split the population, we've separated the population, and that prevents the groups from breeding with each other. And in these two groups, obviously, there will be genetic variation. There will be differences in their DNA originally due to mutations. So there will be some genetic variation. And in the two areas where we've separated this population into like two different parts of the island, or maybe the sea has separated the population, so some are on this island and some are on that island. There could be slightly different environmental conditions. So maybe there are different sources of food where they live. Maybe the climate or the temperature or the amount of rainfall is slightly different. Maybe there's different predators. Maybe there are different diseases. There has to be some kind of difference in the environment. So there's different kind of pressures, different selection pressures, as we call them, put onto those populations. Now, in each of these different environments, we will get natural selection. So individuals with characteristics best suited to each environment are more likely to survive, reproduce, pass on their genes, just like we say in a natural selection question. Okay, a lot of this is similar to natural selection, but you've got to remember that if it's a question on speciation, we have physically separated the population. They have been isolated by a geographical barrier, okay? Obviously, we still say there's genetic variation, but we've got different environments. So we're kind of saying natural selection happens differently. Different characteristics give an advantage. So different organisms survive, reproduce, and pass on their genes. Gradually, over many generations, many rounds of reproduction, you'll get an accumulation of changes. So genetic changes and genetic differences build up in the population and genetic variation increases. And eventually, over many, many, many generations, we can form two new species because they would no longer be able to interbreed to create fertile offspring. They are now classed as separate species because the biological definition of a species is that they can breed to produce fertile offspring. But now, because of the accumulation of genetic differences, they can no longer interbreed to create fertile offspring. And we say speciation has occurred. This type of speciation is called allopatric speciation. You don't have to remember that for GCSE because it's the only one we learn about anyway, but you do have to remember the steps and you can kind of learn it as a model answer, right? So you have to have isolation. There has to be geographical isolation, a physical barrier that separates the population. You have to say that in each population, there will be some genetic differences or genetic variation due to mutation. There has to be differences in the environments, so natural selection will occur differently. There has to be an accumulation of genetic differences, and eventually you say you form two new species because they can no longer interbreed to create fertile offspring. We've got two new species arising from a pre-existing species that was separated or isolated.
I hope you enjoyed that video. Make sure to like and subscribe and click the bell icon to turn on notifications so you don't miss out, because we're going to be posting more videos just like this very, very soon. Don't forget all of those links in the descriptions. You can get your free flashcards by filling in your details. We'll send them to you straight away. You can also find the link to access my resources for everything you need for all of your GCSE revision. And you'll find the link there to join our exam masterclasses to revise with us before your GCSE exams. See you in the next video.